Yes.
Then I would like to welcome Cyril Pommier from French National Institute of Agricultural Research. So maybe you just mentioned the agricultural.
Really personal.
Okay. Also, please introduce yourself.
Yes, of course. That should be right. Okay. Thank you. Okay. So it's all right? Okay, perfect. So hello, and thanks to the organizing committee for inviting us to present the data management in PHENOME. I'm saying us because we are sort of two people talking here. First, Cyril Pommier, myself, and Pascal Neveu. We are both leading the information system for PHENOME. And we are both computer scientists, engineer, and researcher, and tightly related to phenotyping research in France and in Europe. So I will present you the scientific context of PHENOME, what is exactly this project, and why we are dealing with really big data. So INRA, it's a French Institute for Agricultural Research. It has three main topics: so agriculture, environment, and food, or the chain from the field to the consumer. It employs 10,000 persons, among which 4,000 researchers, and it's spread among the 18 centers in France. So, first question, since we are dealing with phenotyping, what is phenotyping? It's simply what we are measuring, observing, assessing when we study the interaction between the plant genotype and the environment. In other words, it's heritability versus a specific environment. So, what do we measure when we are interested in phenotyping? First, the environment, especially climate, pathogen pressure, also disease, soil physico-chemical capacities, specificities, moisture, tension, and so on. We are very closely looking at the architecture of the plants, their structures with leaf area and so on, the biochemical content with chlorophyll or protein content, and the state, the fluorescence, the skin temperatures, and so on. So in PHENOME, we are not simply dealing with phenotyping, but with high throughput phenotyping. Indeed, traditional phenotyping, it's free text numbers, assessed, observed directly in the field or in controlled chambers. And this is inadequate for large-scale computerized comparative analysis. We need to decipher simple trait components, like for instance yield, which is related to disease resistance, to growth kinetic, and so on. And we need to couple phenotype with gene analysis technologies to have very powerful association genetic studies. So why do we want to do so much high throughput phenotyping? It's to be able to give answers to adaptation to climate change, taking into account human health, both for farmers, the ones who are dealing with the plant growing, and the consumer, of course. We need a more efficient use of natural resources, including water or sunlight, in our farming practices. We need sustainable management, biodiversity, and we need food security, and therefore crop performances. So we have got big data management in phenotyping. Well, we are not dealing with zettabytes of data like in astrophysics, but we are dealing with very, very heterogeneous data. This heterogeneity opens new insights for both genotype and phenotype interaction, for cultural practice, genotype and environment interactions. And this allows us to know which theories are consistent and which ones are not. This is also a way to provide us with decision support by combining, analyzing, and predicting several levels of data refinement. So when we want to do genetic association genetic to have better crops, we need to do an analysis of the interaction between genotype and phenotype. The problem is that for the genotype, we've got a real deluge of data. We really have got not all we need, but a lot. There is a high throughput genomic with genome sequencing. It's really mature and it's quite affordable. Now we can sequence even very complex genome like the wheat genome. We've got also genotyping, which allows us to access to the variability at the individual level. But despite all these advances, phenotyping is still very time and resource consuming. Phenotyping is still, today, a bottleneck for both plant science and breeding, for now. Indeed, there is high throughput phenotyping, which is quickly developing, not only in France, but all around the world. One of these initiatives is therefore PHENOME. It's a national project, a French project. Its objective is to build an infrastructure for high throughput plant phenotyping based on existing platforms or evolution of non-existing platform. It is also focused on developing a suite of methods for having innovating way to assess phenotype, develop and implement those methodology both through material, sensors, data management and data analysis. And PHENOME is also integrated in international networks, namely EPPN, Transplant, and POD with Australia. So, PHENOME, it's 9 platforms across France. You've got 2 control platforms, 5 field platforms, and 2 high throughput omics. The 5 field platforms, they are dealing with a high variety of spatial-temporal scales. We are assessing phenotype at the cell level, at the organ level, a whole plant, the canopy, the community, a whole group of plants. We are also dealing with very heterogeneous data types through drones or automated phenomobile, like the one you're seeing down on the left. We can produce a lot of images at several wavelengths. And we still are keeping, of course, human readings. We also got control platform, which have the possibility to control the environment and to automate totally the observation of the phenotype on the plants. It also have various spatial-temporal scales and data types. And last but not least, we've got 2 omics platforms in which samples taken from field platforms or control platforms can be assessed for structure of biopolymers of the seeds, for instance, or metabolomic phenotyping, simply knowing how much, what is the quantity of different molecules in the sample. So some figures. In 2014, we are only beginning to really build PHENOME. Not all platforms are there. Nonetheless, we have produced 20 data sets in high throughput plant phenotyping in fields or in greenhouses. Each experiment gives 1 data set between 2 terabytes and 10 and even 14 terabytes. This is, for instance, 7 million of records in database, plus more than 1 million of RDF triplets, plus 5,000 of images. That's a lot of data, and the problem is that we really have to keep all of it, especially the raw big data. So we are considering that we will produce 100 terabytes of data by years, even more for the upcoming 10 years, and that we will need to keep and archive those data. So here is the scientific context. So now, what are the needs, the challenge, and the solutions that we have proposed? So we've got heterogeneous constraints. We need both to be able to do real-time acquisition and analysis of data. And on the other hand, we need to be able to provide a long-term integration, data mining, and archiving. We need to be able to preserve on the very long term, 10, 20, 50 years for the raw data. So it's petabytes to store for a very long time. We really have a big challenge on identification of the plant material, the varieties, the experimentation objects, the scientific variables, and so on. And we need to ensure long-term data access through open data capability and the use of digital object identifier. So, volume growth, it's relative, but it's really growing year after year. We are producing more and more data. We need to have this real-time acquisition and treatment. We need to be able to assess the quality of the experimentation day by day to be able to conduct the treatments and the next observations. So this is therefore a challenge for both storage, management, and analysis and long-term archiving. For this, we need a high-power computing system, so HPC, and storage. For this, well, we could have used purely internal capabilities, but we want to go more to a cloud approach, which allows us to have access to on-demand infrastructure and elasticity. For this, to be able to do this, we need to be able to set up virtualization technologies. So all of our tools, of our analysis pipelines must be able to run in virtual machines. We are also developing them with a database parallelism approach, the same operation on different data. And those clouds, on the beginning, will rely on data centers like the CNES or some INRA data centers, and grid like France Grid, clusters, and so on. But it will be a network of different institutes. And last but not least, once again, we need to construct a strict and sustainable data life cycle and data long-term archivage. So we've got the problem also of data heterogeneity, with extremely diverse data type. Those data types can be associated to different levels of objects inside an experiment. It can be a population, can be a species, an installation, a site, an observed plant, even an organ or a specific leaf of a plant. It can be comparative, a mutant versus a wild type, or absolute, days to flowering for a cultivar, for instance. And furthermore, there is also a big challenge for interoperability, because the object in our experiment must be very clearly linked to other types of data, seed stock, genes, experimental method, and so on. So the challenge for us is to find a good way to represent in a consistent way across experiments, research, and communities and species what is an experiment in all its richness. So for this, we've begun with the more crucial problems, the naming convention and the identification of objects. For the plant material, the experiment and the experiment elements, technically it's rather simple. It's the use of URI for naming, addressing, and identifying the object. It is a standard approach, it's existing, it's compatible, it has been used in real life, so we know it's working. So it's a good way to do, and furthermore we can encode them in a QR code, which is very important for us for field experiment. Another important thing is the use of controlled vocabularies, semantic interoperability relations, and metadata, therefore ontology sets. This is highly important for trait and environment definition of measurement protocol, which can vary a lot from one experiment to another. This is a hard traceability to set up, and the main problem here is not really technical, is to get all people to agree on something enough compatible. There is also ontologies for plant description for both anatomy and stages, like the plant ontologies, which has been existing for years now, species-specific ontologies, and application ontologies, which allow us not to set up only data quality and data comparability between data sets, but to ensure the quality of the process of the experiment. Through application ontologies, we can be sure that a specific observation, for instance, leaf area index, has been done only after a specific treatment with a nutriment and so on. So as I said, the identification of ontologies and of objects, it's a lot a work of coordination among people. The example of plant material is very revealing. In our current projects, PHENOME or other companion projects, we've seen that we need a coordination of recognized authorities for both synonym handling, for getting a unique identification in PHENOME and outside of PHENOME, and prevent duplicate ID for the same material. This is a very important point. We could handle an infinite number of synonyms, but it will lead us really to big problem to integrate data in consistent data sets, and especially if you've got hidden duplicates, like the one in this example, the PHENOME CF1235, which is probably the same as the Eurisco one, but without explicit deduplication. We've got no way to find it. So to have those recognized authorities for this deduplication, in France we've got biological resource centers. There are seed banks at the European level which are providing the same service, among a lot of other things. And there is an international references, and especially Eurisco. Another important thing for the ontology building is that our colleagues for phenotype have been using and developing phenotyping variables for years, and they don't want to lose all this work to stick to an international reference ontology. We've got to do a bottom-up approach to go from their variables to the emerging standard from what they are really using. So for the information system, for the database, strictly speaking, solution, we had to set up a distributed, layered system with two main components, one for the data production with the local information system, and the other one for data integration on the long-term approach, which is therefore necessarily distributed. So the local information system first, it's responsible of the day-by-day data acquisition, of the analysis in real time for experiment piloting, and of the first level of heavy analysis of the data produced at the end of the experiment. There is, of course, an important layer of data security, namely reliable storage with reliable backup, which is necessary at this level. The second layer is, well, first we've got to have FIS, the local information system, in all the 9 platforms of PHENOME, and all those platforms have to be able to dialogue to exchange data among each other. It can be simply the identification of the experiment, the identification of the plant material, and so on. For this, we are developing a common layer of web services. Well, we've called it web services, but it's both an API, an RDF system, and an indexing system based on Lucene Solaire for speed. And since we are now in a distributed system, we need identification and ontologies. Yes, okay. And on top of this web service, we can provide a better visibility to the data. First, with an integrative portal, EPHASIS. Secondly, through EPHASIS, through GNPIS to European portal, to European project like Transplant, and with IS, which is not only European but international. We can also provide free access to the web service layer to other projects like EPPN or other. It's not working. Okay, better. So, on a technical aspect, FIS, the local information system, is based on a relational database. It's a simple and generic model. And on RDF triplet store with the Sesame triplet store. It's therefore a combination of semantic web and relational database plus a file system for the images. It is also integrated with the analysis tool for interoperability and integration, with example like OpenALEA, which is developed between INRA and INRA, or Skiflower, which is developed in France. The integrative layer, so EPHASIS, is based, as I said, both on semantic, RDF, and indexing technologies, a bit like the approach taken in vitro and vivo, which are a portal for publication, which are deployed in the US. Therefore, we have the possibility to have open data and open API on the whole PHENOME information system. One of the last challenge of PHENOME is data cleaning. We must ensure real time that what we are producing is consistent, that there are no duplicates, that there is no breaking in the kinetic curve, and so on. For this, there are specific tools which are developed. We need to find also these guys missing data, outliers, and so on. The specific tools developed include unsupervised curve clustering, curve fitting over dynamic constraints, and clustering of image histograms. In conclusion, for PHENOME, we are dealing with high throughput phenotyping data. It's very hard to produce. It's almost as hard to manage, and it's also hard to analyze. Thank you.
Thank you very much, Cyril, for this introduction also into phenotyping and development. This is also one part my group is involved in some phenotyping projects where we also do hyperspectral UAV imaging and those things, so that we face in a smaller scale very similar problems. So are there any questions? Yeah, there is one.
Ask my favorite question. What's the long-term perspective? This is now a project, right? So, or is it planned to run for the next 20 years?
Yes, it's a very long-term infrastructure. It's been initiated by a project for first funding, but what we are committed to do is to gain financial stability.
So that means that some users will have to pay pay at some point for
They have to pay all the funding. Yes, yes, already now, because all the funding is on the development of the platforms, of the robots, of the drones, of the new sensors, of the new methodologies.
I also have a question. How open is the data? I believe that if the outcome of such a phenotyping project is that maybe the best winter wheat phenotype is found, this could be quite a lot of money for the project or company who developed that. So is it really open?
So, as I said, it's an infrastructure. So whether the data is open or totally private is up to the customer. So among the customers, there are public projects, there are private public projects, and there can be someday private companies. So public-private projects someday will release their data. It's up to the consortium, and of course public data, it's immediately public.
So I do not see any other questions, so then I would like to thank all the speakers of today. And
Thank you.